Microalgae are powerhouses of nature. They exist in almost every region on Earth and can survive temperatures up to 70 degrees Celsius. Not only that, microalgae are photoautotrophic, which means that they can produce their own food through photosynthesis, just like plants. Even better, in the process, they absorb carbon dioxide, which actively cleans up the atmosphere. This means that they contribute to stopping climate change. But can microalgae be made even better? Let's find out. Microalgae are incredibly useful organisms, as they can be used to produce a variety of products, including biofuels like biodiesel and bioethanol, important substances like glycerol, and nutritional products such as vitamins and amino acids. The best part? All these products are carbon neutral. Because algae produce energy and grow through photosynthesis, they absorb carbon dioxide. Even if that carbon is later released into the atmosphere, the net amount of carbon is still the same. This makes microalgae-based products very sustainable. If microalgae are so useful, why aren't they being used today? It seems possible to use microalgae to generate carbon-neutral biofuels, for example. One of the major issues with microalgae is how they are grown. Algae need to be grown in a structure called a photobioreactor, which allows the mixing of nutrients and water, along with access to sunlight. Unfortunately, these are really expensive, making microalgal products less profitable. But what if we could augment algae's powers so that they could produce more useful goods? Well, we can, with the power of gene editing. Broadly speaking, gene editing is a method of editing the DNA of organisms. DNA is like a recipe book for an organism, and contains all the necessary information for it to grow and reproduce. Gene editing is like writing in this book, changing the recipes inside. By editing the DNA, we can change how the organism behaves. Gene editing tools like CRISPR-Cas9 allow us to do this. In this video, I will cover some of the tools that are used for gene editing in microalgae, allowing us to make carbon-neutral products a reality. If you wanted to edit the genes of microalgae, how might you go about it? One starting point might just be to randomly cause genetic mutations within an algae population. Just throw some chemicals at the algae, which will cause its DNA to change, and hope for good results. This may sound like a questionable starting point, but when it comes to microalgae, it's not actually a terrible idea. The sheer number of microalgae in a culture means that if every alga changes differently, the odds are high that there will be a few algae with good results. Random mutagenesis, as it is formally known, is useful when you want to make minor changes in algae that are easy to screen for. For example, it is possible to reduce the number of chlorophyll in algae using random mutagenesis. This improves microalgae's ability to grow in high concentrations. Random mutagenesis is a useful technique for making simple modifications to algae, but it won't work for the more complicated ones. If we want to, say, change the color of the algae, we will need to target a specific part of the algae genome that controls the algae's pigments. Randomly mutating the genes has a low likelihood of producing the specific change that we want. This gives rise to the method of targeted mutagenesis, where we target and mutate a specific part of the algae's genes. One of the best ways to perform targeted mutagenesis is to harness another microbial superpower, CRISPR-Cas9. CRISPR evolved as a defense mechanism of the microorganism Streptococcus pyogenes. It employs a special protein called Cas9, which allows it to make cuts to the very DNA of organisms. Scientists harness Cas9's cutting ability to edit the genes of all kinds of plants and animals, including microalgae. Cas9 is one of the best methods for editing algae, as it is cost-effective and easy to direct to specific target genes. It can also be used to make multiple edits to the genome at once, a technique called multiplex editing. Cas9 can make changes to DNA in two different ways. The first is to mutate a gene to the point where it no longer works, thereby stopping its function. This method is called gene knockout, and is analogous to ripping a page out of the algae's recipe book. First, we use Cas9 to make a cut at a target site within the algae's genes. This is similar to ripping a piece of paper in half. The algae then tries to fix this cut, as if you tried to tape the two pieces of paper back together. But the paper isn't the same as before, and neither is the algae's genes. In trying to tape together the ends of the DNA, the algae will inadvertently add or remove a few bases, which stops the gene from functioning. By causing a break in the DNA, Cas9 has led the organism to mutate its own genes, accomplishing our goal in the process. The second type of change allows us to insert our own set of bases into the algal genome, similar to writing your own recipe for the recipe book. The process starts the same way, with Cas9 making a cut at the target site. However, This time, we also insert a set of synthesized bases that hover around near the target site. When the algae go to repair the break, they spot the nearby hovering bases and amalgamate them into the genome. The organism now effectively has a new gene in its genome. Going back to our color changing example, the new gene we inserted might instruct the algae to produce more carotene. This compound, which is what makes carrots orange, would turn the algae orange as well. So, these two methods leverage Cas9's cutting power to make specific edits to the algal genome. But one question still remains. If we want Cas9 to make cuts, how do we get it from out in the lab to inside the algae? And for this, there are two methods. To start, we need to discuss how Cas9 actually operates. Cas9 is a protein that cuts DNA, just like a pair of scissors. But how does Cas9 know where to cut? In the same way that scissors are guided by the hand of the user, Cas9 is guided by PAMs and guide RNA. PAMs, or protospacer adjacent motifs, serve as a launching point for Cas9 to bind to the DNA. The PAM is a sequence of usually 3 bases, often of the form NGG, where N is any other base. A PAM has to be found somewhere near the target site in order for that site to be used. Cas9's second guide is guide RNA, special sequences of bases that match the area surrounding the cutting target. When Cas9 approaches a target site, it will first check that the guide RNA and the surrounding DNA match up before making any cuts. Guide RNA has to be synthesized specifically for the target site, which means that it also needs to be attached to the Cas9 protein at some point in the process. There are 2 options: assembly of Cas9 and guide RNA within the algae, in vivo assembly, or assembly outside the algae, in vitro assembly. Let's use an analogy to better understand the difference between in vivo and in vitro assembly. Think of Cas9 like a robot, and the guide RNA as the program that will instruct the robot on where to go. The robot's goal is to retrieve something from a building. With in vivo assembly, the robot will receive its program only after it lands within the building. Likewise, Cas9 only joins with the guide RNA after they both make it into the algae. Alternatively, the robot could receive its instructions before it goes near the building. This is analogous to in vitro assembly, where Cas9 and the guide RNA are joined together before being inserted into the algae. The difference between in vivo and in vitro is the crux of the difference between the 2 ways to deliver Cas9 to the algae genome. The first method for getting Cas9 to the algae is to use a vector, also known as a plasmid. A vector brings together Cas9, along with a bunch of other parts to successfully deliver Cas9 to the cutting location. Essentially, a vector is just a long string of parts that can be inserted together into the algae. With a vector, we use in vivo assembly, which means that Cas9 and the guide RNA are brought in on the same vector, but on different parts of it. They will later be joined within the organism. An important part of vectors is the selection marker. This is essentially a harmless gene or dye that will activate when it reaches the algae's DNA. For example, some experiments will send an antibiotic along with Cas9 into the organism. Then, they will infect the whole culture with the virus associated with that antibiotic. The organisms with the antibiotic will survive, while the rest will die. The ones that survive are the same ones that Cas9 successfully reached, since the antibiotic was attached to the same vector. This allows scientists to figure out and select for which algae were properly edited. Sometimes, vectors will also use a promoter to control when Cas9 is activated. This is similar to putting our robot from before into sleep mode. When we press a button, the robot turns on and is able to perform its task. Similarly, by activating or inducing a promoter, Cas9 will be turned on, and will then proceed to follow the normal cutting process. With our vector fully assembled with Cas9, guide RNA, selection markers, and promoters, we're ready to deliver the vector into the algae. And I'm going to show you one of my favorite techniques for doing this: agrobacterium infection. Agrobacterium is a type of virus you might be familiar with if you've ever seen one of these trees. The trees in the photo have a tumor-like bulge on them, which indicates that they have been infected by agrobacterium. Agrobacterium is a parasitic virus that invades plants and takes over their systems. They essentially turn the plant into a virus-making factory by delivering in their own DNA. This is as if agrobacterium had hacked into the plant and taken over the system. Why is this related to Cas9? Well, since agrobacterium is so good at taking over a plant by bringing in its own DNA, what if we could have it transport in something that we want in the plant? Or in this case, the algae. By replacing agrobacterium's infectious insertions with our own vector, and then inoculating the algae, the vector is transported into the algae without any complications. To me, this is incredibly cool, that scientists are able to harness an existing type of algae-invading system and put it to use. CRISPR vectors are one excellent option for delivering Cas9 and guide RNA into the algae, and then assembling them in vivo. They employ selection markers to screen for editing success, and promoters to put Cas9 in and out of sleep mode. In order to get Cas9 into the algae, we harness agrobacterium to deliver the vector. The alternative to vectors is the opposite in vitro method, known as the RNP method. RNP refers to the Cas9 ribonucleoprotein. As mentioned before, we can use in vivo or in vitro assembly to join the Cas9 and guide RNA. RNP uses the in vitro method. Here, Cas9 and guide RNA are combined before being delivered into the algae. Generally, the RNP method is simpler to execute, and can often be more effective than vectors in algae. However, this is a developing field that requires more research. Ribonucleoproteins can be delivered through a process called electroporation. Electroporation is particularly effective over agrobacterium when the algae species does not have a cell wall. Electroporation first places the algae and the RNP in one area, and then uses electricity to force them together. The algae are forced to absorb the RNP, which allows Cas9 to enter the algae and begin doing its job. These two methods, vectors and RNP, form the current toolbox for CRISPR-based gene editing in algae. It's worth noting that this form of gene editing is very much in its infancy in microalgae. Many of the techniques that I've talked about have only been demonstrated with proof of concept. There's still a lot of work and research to be done before CRISPR-Cas9 can be used frequently in algae. This video has covered a lot, and if you're still here, well done. Let's summarize. We started by discussing the power of algae, and how they can make their own food through photosynthesis and can be used to make carbon-neutral products. We then went on to discuss the ideas of random and targeted mutagenesis, and the use cases of each. Then, we went in depth on one method of targeted mutagenesis, CRISPR-Cas9, a protein that has the ability to cut DNA, causing genetic mutations. Lastly, we talked about the two main delivery methods for Cas9, vectors and ribonucleoproteins. What are the implications of all this? Well, recently, scientists have been testing out different types of genetic modifications in algae, and the successful trials have been rolling in. It has been shown that we can increase the production of fuel in algae by over 70%, and improve algae's growth rate by 50% or more. We might even be able to make algae into a carbon-capturing supermachine by improving how algae perform photosynthesis. There's tons of potential to be tapped in algae, and in the near future, gene editing will be used to unlock it. I hope you enjoyed this video, and if you did, you may also enjoy checking out some of the links in the description. See you next time!